Hello everyone, I'm Shubhashini Vijay Kumar. The topic that I get is translation, genetic basis for protein synthesis. Now we'll look what is translation. Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules including mRNA, small and large subunit of ribosome, tRNA, and lastly go for release factor. This is process broken into three stages, which is initiation, elongation, and termination. Now we look into the process. This is the eukaryotic mRNA, the substrate for translation as unique 3' end called poly A tail, and how it's look like. The mRNA also contains codons, the colorful thing in the video that's known as the mRNA codon, which is specific for amino acid. A methylated cap is found at the 5' end. Firstly, we start with the initiation step, which begins when the small subunit of ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule which I said above. It contains an anticodon that is complementary to the mRNA codon to which it binds. First, mRNA codon is typically attached to the end of tRNA is corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of ribosome now binds to create the peptide of P side and the amino acid or A side. Firstly, tRNA occupies the P side. Secondly, the tRNA enters A side and is complementary to the second mRNA codons. The methionine is transferred to the A side amino acid, the first tRNA and then axis. The ribosome moves along the mRNA and next tRNA is enters. This is the general and basic step of the elongation. We can see clearly in the video. As elongation continues, the growing peptide is continually transferred to the A side tRNA. The ribosome moves along the tRNA and new tRNA enter. When stop codon is encountered in the A side, a release factor enters the A side, which is the purple color one. We can see clearly in the video. It enters the A side and translation is terminated. Here the termination process occurs. When the termination is reached, the ribosome dissociates and newly formed protein is released, and it's also known as peptide. Move on to Garrett's experiments on genes and enzymes. The reason why we should learn about his experiment is because he is the one who first suggested the relationship between genes and protein production in early 90s. He got this idea when he studied patients who had the inherited disease alkaptonuria. Everyone thinks right what the disease is about. Here we go. Patients with this disease accumulate large amount of homogenetic acid which causes the urine to appear black. This trait is inherited through a recessive pattern, and also this disease is due to a defect in phenylalanine metabolism. From this, Garrett described this disease is an inborn error of metabolism, and he also suggested that the inheritance of the trait was associated with the production of an enzyme, which in this case was defective. The picture shows that the metabolic pathway of phenylalanine. That's all from me. I give you guys a quick overview of translation, the genetic basis of protein synthesis. I hope everyone understand. That's all from me. Thank you very much.